Good evening, everyone, and welcome to the Real Science Exchange, the pubcast where leading scientists and industry professionals meet over a few drinks to discuss the latest ideas and trends in the world of nutrition. Today is day 2 of the future directions in Choline Symposia here with the University of North Carolina, and I have 2 guests with me. First is Dr. Jonathan Bortz. Jonathan is the VP of Nutrition Science at Balchem, and Dr. Eric Ciappio, he's a manager of Nutrition Science also at Balchem. Gentlemen, we had a great day yesterday here at the symposia. Learned a lot. Eric, the first day was all about choline and prenatal nutrition and choline and early life and childhood nutrition. What were some of your key takeaways from day 1?
You know, I think for me, it's just so incredible to see how far the field has advanced. Even in my time with Balchem, we're fortunate to be in a world now where there's been a lot of randomized controlled trials showing the benefits of choline supplementation during pregnancy and how that impacts offspring development. So what we saw yesterday at the sessions was the benefits of maternal choline supplementation in terms of baby's cognition, both immediately after birth and persisting up to 7 years in life. Improvements in DHA status as well when choline and DHA were given together. We also saw a lot of incredible data on the ability of choline to support what we call fetal resilience, its ability to sort of adapt and deal with external stressors, be it emotional stressors that mom faces, be it exogenous toxin stressors that mom may consume. The abilities that choline seems to demonstrate in early life are really remarkable. We also had a good discussion on choline and lactation and how some of the specific metabolites of choline were present in breast milk and some of the differential effects of that, which is an area that does not get enough attention, sadly. So just a lot. We also had a good discussion on the role of choline in food aid and how adding choline to certain food aid programs might benefit in terms of areas of severe acute malnutrition. So a really broad ranging, and we really took the charge of trying to identify the role of choline in early life nutrition broadly, and I think that's reflected by its really varied roles and the varied clinical trials that are out there. So really, really exciting day yesterday.
Yeah, it's a great day. Just as a reminder to our audience that we released day 1's podcast last week. Please go back and listen to that one, full of great details and information there. Dr. Bortz, day 2's coming up. We're going to shift gears just a little bit, and we're going to be talking about choline and adult nutrition. What's some of the things the audience can expect to hear from today?
Well, Scott, first of all, let me say this. About 5 years ago, when some of the very exciting information was being developed and generated by our clinical investigator collaborators about the benefit during pregnancy and children and so on and so forth, I remember having a meeting with some potential customers who were impressed with all the data, and when everything was said and done, they turned around and said to me, well, what about us? Mm-hmm. And what that did was it opened my eyes at that time to say we really needed to expand our vision into childhood, young adulthood, and beyond adolescence, and into the benefit for mature humans. And I think what we're going to see today, with the focus yesterday being on the prenatal, early childhood and childhood, what we're going to focus on are some of the challenges that we see in our health environment today, with obesity, with the development of really a silent epidemic with fatty liver, with cognitive decline. So we've invited really the world's leading investigators in these areas to come and share with us their science and their literally breaking news, how they're looking at things. So I'm anticipating a very exciting day in a new chapter, and that is how it affects, how choline affects all of us from prenatal all the way through to old age.
Yeah, great summary, great way to kick off day 2. Looking forward to it. Thank you, gentlemen.
New research is changing everything we thought we knew about choline's impact on the cow and her calf, and top scientists have a lot to say about it. They are presenting new research that supports choline as a required nutrient to optimize milk production, choline as a required nutrient to support a healthy transition, choline as a required nutrient to improve calf health and growth, and choline as a required nutrient to increase colostrum quantity. This new research is solidifying choline's role as a required nutrient for essentially every cow, regardless of health status, milk production level, or body condition score. Learn more about the science that is changing the game and the choline source that is making it happen. Reassure precision-release choline from Balchem. Visit balchem.com/scientists say to learn more.
So our next guest is Dr. Mark Manary. He's a professor of pediatrics at Washington University School of Medicine in St. Louis, Missouri. Dr. Manary, you gave a presentation, or will be giving a presentation later today called Choline and Food Aid. Can you start off by telling us just a little bit about food aid and what that is and does it entail?
Well, food aid products are specially designed to address needs from crisis situations. So, right now we have a huge crisis in Gaza, and so what's needed there are these specialized food aid products. They're standardized, okay, to meet needs of great deficiency or inadequacy. And so on the most extreme side, we've got something called ready-to-use therapeutic food for children who are starving to death. And we're not talking about people who are poor or don't have shoes or may not be eating like they should. We're talking about people that face a serious compromise, even death, in a few months if they can't get the proper food. So that's one of the food aid products. And then for children who are just morally underweight in the process of starving, we have other food aid products. Choline is not prescribed in any way in these products. And as an essential nutrient, choline seems to be kind of, always a lap behind many other nutrients, in catching up. And so our goal of this investigation is to place choline in food aid and say, hey, what happens?
Okay, so it's kind of a research project then, is what you're doing with food aid.
It's a clinical trial. So in other words, thousands of children who are receiving this food aid will be given either food aid with choline or the standard food aid.
Okay.
And we'll measure, in some cases, cognition. In other cases, how well they recover.
And where's this research taking place?
This is going to take place in Malawi, a place that I've been. So parenthetically, I'm introduced as someone from St. Louis. But where I work is in sub-Saharan Africa.
Okay.
And that has been the case for a really long time,
okay.
1985. And so I have 4 teams, each in different countries, about 200 people who work for me. And we're doing trials, and food aid is one of our primary foci.
Okay. And so when you put this trial together, what's your hypothesis? What are you anticipating that you'll see?
I anticipate that we will see, we saw a 6 to 15 IQ point difference by adding fish oil, or DHA. And when we combine that with choline, I'm expecting we'll see a doubling of that.
And is that a certain age that you administer the choline, or does it matter?
It's administered when they're severely malnourished.
Okay.
So it isn't a well population. It's a population that's starving to death, and typically most of those kids are under 2, but they're almost never under 6 months.
Mm-hmm. And so your presentation is going to be basically just kind of outlining the research that you're doing and the expected outcomes.
Yeah, and talking about the stuff we've done in the past.
Okay.
So a trial with DHA. One of the really exciting things about doing trials like this that are in the vernacular, they would say well-powered, which means that there's thousands of participants, which means that the outcomes of any 10 participants can't really bend the data one way or another, is that then you're in line to change policy.
Okay.
So what we did with the DHA trial was we took that to the WHO, then we took it to Codex Alimentarius, so that is the FDA of the world, if you will, the international FDA. And now every child who gets ready to use therapeutic food has to have DHA in it, or the balance of the kind of fatty acids that constitute DHA, omega-3 polyunsaturated fatty acids.
And so, boy, I had another question here. Go ahead, Tom.
Yeah, no, thank you. Dr. Manary, I have an interesting question based on, we've seen, and actually we'll see from other presentations here at the symposium, that there's definitely a prenatal impact of DHA and choline combined in terms of providing greater DHA status or more beneficial. Do you think that's true postnatally as well? Does that hold firm, or?
I don't know. I mean, we were talking about the trial just now having to do with children, but we also are doing a trial with malnourished pregnant women right now in Sierra Leone. We're adding DHA and choline to malnourished women. Okay, so women, not all pregnant women, but just the ones that are starving, and looking at cognition in the babies. I mean, certainly it's a hard. Do I have an opinion on whether this is just a developmental issue? I think is what you're asking. So if you don't get it at the time development's going on, versus an ongoing issue, I'm going to guess that it's an ongoing issue. Choline's the kind of metabolite that's used every day for everyday work of the brain, and so having periods of your life when you don't have choline, I imagine if you then added it back into your diet, you would see better performance or something. Okay. Whereas I don't think you can have things that happen during very formative points in your life, those like being in the womb, being in the first 6 months of life and so forth, that stick with you forever.
You know, one of the questions one of the statements you made is that your research, you're hoping that it'll help direct policy. Now, is that going to be policy just for malnourished children, or is this going to be broader and looking at other children that are not necessarily severely malnourished?
No, I mean, the policy we're talking about has to do with what is in food aid products.
Food aid.
So food aid products have a certain, specification. Mm-hmm. And you can't say that something is RUTF when it doesn't meet these 335 specifications. And choline we're hoping is going to be one of those.
Okay. All right. Very well. Tom, anything else?
I think one of the things we've seen, we talked a lot about cognitive, is do you think there's going to be benefits? Because one of my understanding is that one of the consequences of low choline is potential liver problems. So do you see that as something that you're looking at, or?
We're not specifically looking at it. I mean, we have very strongly believed that fatty liver will be diminished with choline supplementation. The populations we work with in sub-Saharan Africa just really aren't obese at all.
Right.
And so, why we say, fatty liver is such a big problem going up in our society has to do with obesity.
Right.
Yeah.
Okay.
Yeah.
Very well. Appreciate you joining us today. Just kind of one final question. Would you mind kind of giving us just one key takeaway for consumers out there? One takeaway from the research that you've done, whether it's DHA with choline, what would be one message you'd give them?
Well, I guess the message I would impart for consumers is that getting ample amounts of choline from commonly used foods, particularly plant-based foods, is difficult. And so there has to, the consumers need more help in how to achieve that.
very well. Maybe just kind of as a follow-up that. So it's difficult getting it for plant-based foods. So what are some foods that are perhaps rich in choline that we should be consuming?
So the bugaboo with that particular question is that, yes, there's some choline in milk and there's choline in peanut, but if you had to increase your consumption high enough in those foods to get to these levels that are being talked about next door, that's not realistic.
Got it.
You know what I'm saying? That's why my message was it was tricky to get there.
Yeah.
And I'm not using the word supplement on purpose.
Okay.
but I am intimating behind that it might be something that is better addressed for many people through a supplement.
Yeah. Very well.
Yeah, I think that's a really good point, because what we've seen definitely is anyone who's on a plant-based diet, just like they have to worry about B12, they worry about choline as well. And so I think, it's always going to be food first when you talk about policy, and I get that. But liver just isn't eaten in great quantities anymore, and eggs are often very challenging, for people. So,
yeah, and the populations I work with in sub-Saharan Africa are eating animal source foods once a week.
Yeah. Yes,
that kind of thing. It's
just
not part of the,
Great.
This has been a great discussion, Dr. Manary. Thank you for joining us today, and thank you for everything you do.
You're welcome.
And we're back with Rima Obeid from Saarland University Hospital in Homburg, Germany. Now, believe it or not, Rima, I'm also somewhat from Germany. I was born in Frankfurt. Did not stay there very long. My father was in the service, but I've got that little bit in common. You gave a presentation earlier today called Choline and Pregnancy Outcomes. Can you kind of just give us a real top-line overview of some of the things you covered in your presentation?
Yes, indeed, we have shown that if choline amount in the diet of the mother is low or insufficient, this will mean higher risk for getting a child with serious birth defects, and this will also affect the liver health of the fetus and the infants.
Oh, that's very interesting. Now, did you happen to follow some of the health impact on the babies after they were born, like the immune function or anything like that?
No, indeed not. But the study that we published is showing that if choline is sufficient during lactation, this will be good for the liver of the infants, the breastfed infants, of course.
Okay. And did you share with the audience some research then that you've done personally there?
Yeah, we have done a recent systematic review and meta-analysis on the association between the dietary intake of the mother of choline and the risk of neural tube defects.
Okay.
Like spina bifida, for example.
Okay. Tom, you have anything?
Yeah, I think just one of the things that we actually talked about with someone else is I think we hear a lot in pregnancy circles about obviously the risk of preeclampsia or in some cases gestational diabetes, these types of things. Do you think potentially, given the discoveries you've had with liver health, could that be an issue for mom too with, having liver issues during pregnancy? Because obviously a lot of that choline is going to the baby.
Yeah, indeed, we speculate about this right now, and there is a biomechanisms or biochemical pathway justifying that if you give more choline, then the risk of diabetes or preeclampsia could be lower, but we don't have concrete studies on this right now.
So more research still needed.
Yeah.
Okay, well, that's interesting.
So along those lines, what kind of research do you have planned for the future?
Well, we for the near future, we are doing more research on neural tube defects like spina bifida and the interaction between choline and folic acid or folate, because folic acid is standard in prenatal nutrients or supplements, but not yet choline everywhere it should be. And we are looking at the interaction between them, so that if the status of folate in the mother is low, then the importance of choline would be even more. So this is one study. The other study is on the relation between the severity of congenital heart defects, which is very common compared to neural tube defects. So we have 1% of the births with heart defects, and this is a real problem with long-term and high costs. So we are looking at the association with low choline in blood of the children, the mother and the father, because we have seen in a pilot study that there is a family pattern, which could be due to some genetic background.
Oh, that's very interesting.
Yeah.
Yeah, I think given the fact that you've been doing a lot of research, it sounds like we know that choline adequacy during pregnancy is really, really important, both to the health of the mom and the baby. If you met a mom on the elevator on your way out of here, what would you say just to her in a few sentences about choline and things you've learned? What recommendations might you have?
Well, indeed, we know that from population studies that in general, women in pregnancy age are not achieving the adequate intake of choline. And that's why I would advise all women to take choline from pre-pregnancy until the end of lactation if they are not able to achieve this through the normal diet.
Right, because that can be a challenge, I know.
Yeah, it is a challenge, especially because the natural sources of choline are rich in lipids or in fats, and this is not the kind of diet usually consumed during pregnancy. You know, women try not to gain too much weight and so on.
Yeah.
So
Great.
supplements could be a good alternative.
That's great.
Yeah, great advice. Just kind of want to follow up with one final question. So what is the recommended rate that you recommend in Germany?
The total intake is currently, it's not my recommendation. I mean, we have, like, the recommendations are set by authorities, and it's in Germany, like similar to the US, 450 milligram per day during pregnancy and 550 during lactation.
Okay, very good.
So that's the minimum a mom should be getting.
That's the minimum amount. What is the meaning of minimum amount? I mean, even for this adequate intake level, we don't have so strong evidence, but at least this amount should be achieved. Yeah.
Right. Okay, great.
Rima, it's been a pleasure meeting you. Thank you for joining us today.
Thank you very much.
You're very welcome.
Thanks.
Thank you.
And we're back with Dr. Susan Smith. Susan is the Deputy Director of UNC's Nutrition Research Institute. Susan, you gave two presentations today. One was titled Choline and Genetics and Cognition. The other is the blanket recommendation. So why don't we start off with the first one? What was kind of the overall thesis of the talk that you gave there today?
Sure. So we know that genetic variance in choline metabolism is important. What people don't realize, you know, we talk about genetic variance, and we think these are rare things out there, and most of them, fortunately, are rare. But in nutrition, they're actually fairly common in our population, and they reflect what the geographic location were we involved in, and what our dietary practices were in that location. Maybe we were within a group that had a high fish consumption, or maybe we had low access to meats, or maybe high access to milk and meat products. So that can capture gene variants and allow them to be spread in that population because of their diet practices. That was great maybe 12,000 years ago, but today, with our modern McDonald's diet, we now have this mismatch between what we were consuming then and what we're consuming now. So we worry about this a lot because we all carry these variants. They can be anywhere from 1% to 10% to 50% in a population. And we're only just now discovering what these are. And they affect a whole range of risk factors. For example, Alzheimer's disease, or heart disease, maybe risk for kidney failure. So we've all heard about these, but a lot of us don't understand or don't know that they also affect our nutritional needs. So that's the avenue that my work is entering from. And we were looking at choline and asking, are there choline variants that affect how powerful that choline is in treating a disease condition? And the situation we were looking at are children who had, unfortunately, prenatal exposure to alcohol, which we know damages the brain. And alcohol is a powerfully addictive product. It's hard to stop drinking. So we hope that we can identify maybe nutritional solutions that will improve the brain function for people who are affected. And a very important nutrient that we already know about for this is choline. And we've talked a bit about in the podcast, and you've chatted with others about how important choline is for brain health. So we asked a little portion of that question and said, in children who have, brain damage from alcohol exposure, can we use choline to treat this? The answer is yes. It's very helpful. And what we did was we looked at the genetic piece. Are there some children who benefit more? And the answer was definitely yes. And so there is a gene variant that affects how efficiently we absorb choline from the diet.
Okay.
All right? And this variant, some people absorb it better, and some people absorb it worse. And it's a common variant. It's in between 5 to 40% of the population in the US. And it turns out that people who had the variant that reduces your ability to take it up, they benefited the most from the choline.
Because there's a bigger gap to fill.
Because there was a bigger gap to fill. That's exactly right.
Right.
And so that was very exciting to see. So now we know who to target this to.
I think what's so encouraging about that to me is, it's hitting it from 2 points. Number one, I think a lot of times people think, well, what you do during pregnancy, it's done. There's nothing you can do. And I think you're giving us hope to say there are things you can do after the baby's born to help improve with brain development, et cetera, and recover from some of those things. But then on the other level, we're going to get more information as time goes on about the specific needs of each person.
Yeah.
Is that something someone might get from like a 23andMe eventually, or is it specialized now?
Possibly. I will tell you that 23andMe is getting most of their information from folks like me.
Yeah. Okay. That's good to know.
So it's out there, and the information base is growing. I don't think they've got this one yet.
Fascinating.
Yeah. So the other piece we then looked at in that question was we said, okay, if this gene really helps fill the gap, then the prediction is that people who aren't getting choline will have poorer cognitive function because that gap isn't getting filled.
Right.
So we went and we looked at that. And to our surprise, that gap exists, but it wasn't just for the alcohol-exposed children. It was for all the children. All the children. And the children who carried 1 or 2 copies of this particular variant had reduced cognitive performance as compared to those children who were born lucky enough to have the other variant.
Wow.
Even though they were all eating the same diet, presumably.
So this kind of ties interestingly into the title of your second, talk, which I know is the blanket recommendation and the idea of, well, how much should mom be getting? Maybe talk a little bit about that part of your speaking.
Sure. We don't have a good answer yet, unfortunately. But we know that it's very likely that all the moms in this study were not getting enough. Just because we know in the US population, most pregnancies are not consuming enough choline. The message hasn't gotten out the way we should. So on average, they're getting maybe 50, 60% of what they really need. So maybe the pregnancies where they have the good, I don't want to call it really a good variant or a bad variant, but the variant that has strong choline uptake, you can muddle through on that, and they're okay. But the pregnancies where they have the allele that, or the variant that isn't so good at absorbing choline, now if you reduce the intake, that's a double whammy. And maybe those pregnancies would have a much better outcome if those pregnancies were able to hit that requirement and hit that target. And we think that's correct from other studies that have been done in humans, showing that when the women consume enough choline to hit that target, that there's significant improvement in the outcomes. So we think some of those pregnancies are probably this variant.
This is really fascinating research, and I think for those of us who are in the field and understand a little bit about choline, it's super exciting. One of the challenges we always face is how do you translate this to a message that just everybody can. If you were to run into a mom, for example, walking out of the conference today, and she were to ask you, what do I need to do with choline? Why should I be concerned? What might your few messages be in layman's terms?
I mean, the best message is that eating enough choline lets your baby achieve its full potential.
That's a great
You
message.
don't want to limit that potential.
Yeah, that's good. That's good. I think we had a really interesting talk when we went to the ACOG earlier in the year, and one of the things was, we basically said to our, as the message on our board, we said, imagine a nutrient that could change your baby's life, but 90% of moms aren't getting enough.
Yeah.
So I think that's really the message we got to get out there.
Yeah. I mean, in pregnancy, of course, it's a really hard time because everybody's messaging you, do this, do that, don't do this, don't do that. This is one where it's so simple.
Yeah.
And it's a huge, if you will, bang for the buck. There's such a large benefit. And the risk from it is very, very, very small. So it's a can't-lose situation.
Yeah.
It's great to hear.
Yeah, Dr. Smith, you're a great ambassador for the cause, and thank you for helping us get the word out there, right? We're doing the best we can, and I'll ask if you will forward this podcast around when it comes out. All
Will
right.
do.
Sounds good. Thank you for joining us.
Thank you for
Thank you
inviting
so
me.
much.
And we're back, and this time we've got two guests and a new co-host. Dr. Eric Ciappio is going to serve in the co-pilot seat on this one. My two guests are Isis Trujillo and Evan Pauls. And Isis, this was not your first time working with us before. I think you've been at the Real Science Exchange before a couple years. No, it might have been pre-COVID even.
Pre-COVID.
Yeah, so welcome back. It's good to have you here. Isis, would you mind kind of just start us off by giving us an overview of the presentation that you gave yesterday?
Yeah, of course. Well, first, thank you so much for having me here. It's a pleasure for me. Yesterday, I talked about how important is choline during pregnancy, and mostly I focus on brain development and eye development, and I share some of our findings in how important is when a pregnant woman is taking enough choline.
That was interesting. The one thing I hadn't heard before was about the eye development. So what's unique about the eye that's different than the brain that allows that helps with eye development and choline?
Well, actually, it's the cells are coming from the same origin. So brain and eye are going to develop from the same cells.
Okay.
So basically, we found this eye development to be happening more or less at the same time that the cerebral cortex, that is the top part of our brains. And we saw that actually the neurons in our eyes that are receiving the light and connecting to the rest of our brain are also very important to receive enough choline.
Okay. Let me ask you this. Is it only allocated to the brain, or can it help with other organs as well as they develop?
Well, that's a very interesting question. And we are not sure yet, but we think that it may have an effect on liver. But I think Evan can talk more about that in adults. But in embryonic development, we think that probably it will have some effect in the liver.
Oh, so that's a great segue to Evan. Evan, give us kind of an overview of your presentation, and then, if you will, kind of talk about liver development.
Yeah, definitely. Thank you so much for having me here. So my presentation focused primarily on looking at this intersection between choline availability and obesity. and specifically the manifestations of some of the associated chronic disease states that we see with obesity, and really trying to get to an understanding of can choline and its metabolites help ameliorate some of the symptomologies of obesity. And one of the main ones that we are focusing on right now is non-alcoholic fatty liver disease, or just fatty liver disease. And so at least with respect to liver development, although I focus more on the adult side, and we know it's very, very well documented that giving choline to someone who is deficient in it really helps ameliorate that fatty liver. In development, we know that at least I think there's been a lot of evidence on specifically for the mother, in which if she's not supplied a sufficient amount of choline, not only is she donating a lot of her own stores of choline to the fetus so that it can develop properly, but also she's put at a much higher risk of developing fatty liver. So I think that's a really interesting area and really needs a lot of focus to make sure that everyone has enough choline in their diet.
So Isis, what I love about your work is it feels like in choline for so long it's been a little bit of a situation where the mechanism is described as, there's sort of inadequate choline epigenetics and then some problem occurs, right? But you've really shed a lot of light on that, particularly as it relates to brain and eye development. So can you maybe walk us through, kind of a high level of what you found in these last, 2-3 years about the mechanism of how choline deficiency can impact brain development?
Yeah, actually, what we found is that the stem cells of the brain, and these are very particular cells that are going to give rise to neurons, to all the types of cells in the brain, are very sensitive to choline availability. So what we found is that if we don't have enough choline, if a mother is not eating enough choline, these cells in the baby brain are not proliferating enough. So basically the mom needs to eat enough for the baby to build a better brain. And then these cells are going to proliferate, have a really good pool of this number of cells, and they are going to become neurons eventually. So part of what actually was also his dissertation was to find what was happening, how choline was making those cells to proliferate more. And we found that it's through microRNA, that it's a small RNA that does fine-tuning regulation, and that is what is regulating this pool of stem cells.
So interesting. And so I think another message that I learned about the conference overall, but especially I think both of your sections, was that it's not just important for expecting moms. I mean, there is a significant role of choline as we age. So, Evan, for your work especially, this interaction between obesity and choline deficiency and choline levels, I mean, could you sort of elaborate on that and how that interplay might impact specific aspects of health?
Yeah, definitely. I think one of the big things that we're really focused on, and at least we see emerging in our work, is that this idea of lean mass and fat mass, excuse me. And so we know as we age, one of the large concerns is loss of lean muscle mass. And so one of the things that we've seen, particularly in preclinically in males, is that we have this loss of lean mass in these preclinical models that aren't consuming a sufficient amount of choline. And so particularly in our older population and as we age, we're starting to think that perhaps making sure that we have sufficient amounts of choline, perhaps having fairly high levels of choline intake may help prevent that loss of lean mass.
Wow. So one of the goals that we've had for this conference overall is trying to create just short messages that we can sort of communicate externally. So maybe what we can do is, Isis, if you wouldn't mind giving a 2 to 3 sentence summary of just kind of what the state of the union is as it relates to choline in early life nutrition, and perhaps Evan, you could do the same for, let's just call it adult nutrition overall.
Mm-hmm.
I will say that if a woman is planning to get pregnant or is pregnant, needs to eat enough choline to make a baby with better cognition. that will be a very straightforward recommendation. The other thing is that it's important to consider also during nursing or lactation to keep consuming choline, because the development of the baby even occurs the baby's brain occurs after birth. So keeping the breast milk with enough amounts of choline is going to be very important.
Of course.
I think like one of the really interesting things that I didn't touch on too much in my talk, but I had in sort of the backslides a little bit. A lot of evidence is emerging that, what is a diet to maintain health, right? And so a lot of people are looking for a lot of, perhaps you could say like quick fixes, but ultimately there is nothing that is going to outweigh a healthy lifestyle. And really what we think of is really high diet quality. And so we are surrounded in an environment of a lot of processed and ultra processed foods. And so what we really need to focus on is just having a really high diet quality and ensuring that we get that high quality, and that high quality diet is including choline, because choline is a part of that really high quality diet.
Exactly. I think that's what I love about nutrition overall, is there's love and hate, is that there's just 100 boxes that you need to check in order to live that optimal lifestyle. Choline, of course, is an incredibly important part, but it's part of this bigger picture of overall health and diet, and I think you captured that really well, so thank you.
Yeah. I want to thank both of you for joining us today. I failed to mention early on that you're both employees of University of North Carolina here with the Nutrition Research Institute. I believe, Isis, you're a researcher and you're a postdoc, correct?
Yes.
Yeah, that's correct.
All right. Thank you guys for joining us today. This has been great. Thanks.
Thank
you.
And we're back at the Future Directions in Choline with my co-host, Tom Druk. Our guest for this session is Dr. Bryan White. He's Professor Emeritus from the University of Illinois. Now, this week we've been having a 2-day symposium. We're in the middle of that. After the symposium, we're going to have a scientific advisory board, and Bryan's going to be giving a talk to that advisory board. Bryan, why don't you give us just a kind of brief overview, kind of the big rocks of what your presentation is going to entail?
Sure. So the microbiome has been implied into TMAO production, and then the negative effects of TMAO that primarily in cardiovascular disease. That cardiovascular disease side of the story is way outside of my expertise. My expertise is in the microbiome and the gut. And so the question comes, what is the role of the gut in the production of TMAO that gets into the bloodstream of humans? And so there's been a lot of work out of primarily Stan Hazen's lab that's shown a few things that are indisputable. So the indisputable thing is that microbes produce TMA. Yeah. TMA is taken up across the gut wall, and then in the liver is converted to TMAO.
Okay.
Can't dispute that. What you can dispute from the literature is that there's a diet effect on the production of TMAO, and that effect of that diet changes the microbiome in such a way that more TMAO is produced in the bloodstream. Okay. And when I've looked at the literature for this effect, there's a couple of published papers that really were not reviewed very well.
Okay. So
the manuscript in most questions is the Coates 2013 paper, and Figure 2 is really very poorly reviewed, and the data in that I have taken from their small read archive. So I look at the microbiome. The microbiome is a community, and you can ask a couple of questions. You can ask 4 questions really. You can ask who's there. You can ask how many of them are there. You can ask what can they do. That's genetic potential. Mm-hmm. And then you can ask what do they do. Mm-hmm. And the simplest thing to do is to find out who's there and, in a rough sense, how many are there. That's using 16S ribosomal RNA technology, which is what is done in the Coates paper. And so they took individuals given different diets, omnivores versus herbivores or versus vegetarians, and asked who is there and how many are there. And then they tried to correlate that to blood levels of TMAO. And in the paper, it looks pretty reasonably clear that there's a correlation between diet and the blood levels. But if you take apart that figure 2 carefully, and then I took their small read archive, the sequencing that they did of 16S ribosomal RNA, and I did the analysis myself. And I got different results. And my results would be completely opposite of theirs, that there's a diet effect and that there's a correlation between the microbiome in those diets and blood levels of TMAO. So through that exercise, I've contacted the authors. The authors have, in some cases, been cooperative in saying, yes, we can give you data, which is fine. Don't necessarily agree with my results. They said I did different methodologies. So I went back and I did their methodology. Now the problem is that paper was done in 2013. I can do probably 80% of exactly what they did. The other 20%, because databases have changed, technologies have changed, I can't reproduce. But the bulk data does not match their data. There's no correlation between diet, and there's no correlation between TMAO levels in the blood with their cohorts.
Would you say, Dr. White, that it's is it you now have finer tools to analyze, or was it a matter of with what they had at the time, wasn't necessarily drawing the same conclusions, or have you learned things since, I guess is my question.
We have learned things since, but they, to me, and I've been doing this since 1987, before a lot of people were doing it, they didn't present 2 critical pieces of data, or one critical piece of data, let's say. And these are called PCOAs, and they are dots. The dots in space, if you've looked at a microbiome paper, they're the dots in space, and you draw a circle around the dots in space, and you can get 2 distinct sets of dots in space. Those PCOAs are based upon sequence similarity between communities. They did not present those PCOAs. They did a rough statistical model, which you can do on a computer, and it throws out numbers and says they're different. Okay. I never publish a paper, submit a paper, and this goes back to before they submit theirs, without PCOAs. If your PCOAs don't show clear circles, then there's no difference. And when I redid their data and then generated PCOAs, there's no difference. And the statistics show there's no difference. It's a finer level of analysis, but they had all the data to do it. It's literally a push of a button and about 48 hours of CPU time, unsupervised. Why what they say they did not do that, they just used the statistical model. Why they didn't do the PCOAs, I have no idea. But if you saw those, you'd go, nope, sorry.
So it's created a bit of a controversy.
A big controversy.
Yes, and so what are the next steps to get to the bottom of this, right? Because I'm assuming they're saying they're right, and you're saying you're not so sure. And
so the literature since that is mixed as well.
Okay.
I mean, you can find probably as many papers out in the literature that say that TMAO levels in the serum is linked to the biome as you can say is not. Again, the indisputable thing is that microbes in the gut produce TMA.
Yeah.
But can you correlate that to blood levels? And I haven't seen much in the literature that convinces me that the community structure, the presence of those organisms that do produce TMA is correlated or associated with blood levels of TMAO. Mm-hmm. So it's the Hazen group has taken their set of data and they've gone to march with it. And there's plenty of data out in the literature which is contradictory to that, but they're going with their story.
Yeah.
Now, he just recently published a paper, and I'll present a little bit at this CSAB meeting, where he tried to correlate or do predictive modeling of the organisms in the gut and their genes, and try to say, can we use a study where we have cohorts that have high TMAO versus low TMAO, and can we look at the gene content and the organism content in the gut, this community structure analysis, who's there and what's their potential to predict blood levels of TMAO. And guess what? They couldn't. Now, the fallback is you're not looking at activity, which is the last piece of that puzzle, just because you don't have a correlation of the gene content, doesn't mean you don't have a correlation of the activity. I would suspect that they're not going to find that if they look at it.
Yeah.
Well, this has been super fascinating. I think we're going to hear a lot of other talks about TMAO and some of the causality versus the association and all that. But it's fantastic to hear some of this deep dive into the microbiome piece, which clearly is your area of expertise. So
Well, one thing with almost all the microbiome studies out there, causation is the gorilla in the room. And there's really only one disease that has been absolutely correlated to the gut microbiome, and that's C. diff.
Okay.
All the rest of them, there's associations, but the causation root has never been defined for any other disease. So is it the microbiome that causes the disease, or does the disease change the microbiome? And the only one that's been established for is C. diff, out of all the ones that are out there. So
it's great context.
Yeah. We thank you for your time today. Thank you for being a part of our advisory board, and look forward to talking to you later on today.
Thanks very much.
Thank
Appreciate it. Thank you.
Our next guest is Dr. Jonathan Bortz with Balchem. He's the VP of Nutrition Science here at Balchem. Jonathan, you gave a presentation today called TMAO and Choline, Mechanistic Perspective. So give us kind of a bit of an overview of what your presentation was all about.
Sure, Scott. So first of all, in the last several years, there's been attention being paid to some concerns about choline, really advanced particularly by one group of investigators who have claimed that excessive intake of meat and eggs and animal source foods that land up contributing either choline or carnitine land up generating a substance in the blood called TMAO, trimethylamine oxide. And what their hypothesis has been is that this has a negative effect on the cardiovascular system and has been associated with a high incidence of cardiovascular disease. What I demonstrated, or at least tried to share with my audience today, was that we have taken this concern, the safety concern, very seriously, have looked into with tremendous breadth and depth into the studies that have been performed to try and ascertain whether there were some real safety issues associated with choline, which is our product that we market. And fortunately was supported by our senior management to say, explore the science, let the science speak and tell us whether there are issues to be concerned about or not. And I'm happy to say that what I presented were multiple examples of how the concerns about choline with respect to TMAO really cannot be associated, cannot be supported. The claimants maintain that there is a causative effect between choline, TMAO, and cardiovascular disease. But what I was able to present to the audience was that this is really not the case.
Now, is there new research or additional research that's going to be required to demonstrate that, or?
So the answer is some of the studies that initially were published in support of this hypothesis have been repeated and have failed to demonstrate the association. And furthermore, we have continued to look for and support any science that can tease out and evaluate the whole body of information and literature to determine whether there's anything there or not. So there is going to be some ongoing work. We don't believe that this is going to go away overnight, at least the concern, but we are very satisfied that choline does not represent a risk to any users, young or old.
Makes sense.
I mean, for most folks, we've been asking them to summarize sort of the state of the union in their particular area in a couple of sentences. I think you just said that really nicely. I mean, is there anything else or anything else you'd like to elaborate on this area briefly for anyone who might be listening that's, kind of confused by all the stuff that's out there? I mean, how do they make heads or tails of this connection?
Sure. Well, let's a very easy and clear example is that TMAO, which again is the substance that is found in high concentrations in deep sea fish, when a meal is had of cod or halibut or whatever, okay, the amount of TMAO that gets ingested by eating this meal is in the several thousand-fold higher than what would be generated by eating an egg or having a rib steak. So the question is, if this is the case, if TMAO is noxious and toxic to the cardiovascular system, Eric, then why is it that in multiple studies, the consumption of fish has been associated with actually a decrease in cardiovascular risk? So, for example, in a meta-analysis of over 360,000 patients, okay, it's been found that this reduction is nearly 20%, okay, on fish eaters who consume much higher amounts of TMAO than what is contained in food substances that are rich in choline and carnitine.
Yeah, and so I guess what you're trying to say is, or what I'm understanding from you, is that this connection doesn't really make a lot of sense when you consider that fish consumption is really kind of a bedrock principle of dietary guidance for cardiovascular disease prevention, and that doesn't really match with the elevated TMA concentration that's found in fish, right?
Right.
These two things are
Right. It doesn't match at all, and even though, what I anticipate is that when people hear about this, they say, "Well, what's there to talk about?"
Mm-hmm.
And there really isn't anything to talk about. However, this hypothesis has been tied to the microbiome, okay, which is a very sort of sexy and attractive and new age way of looking at disease, and unfortunately, there too, the more intensive investigation of their microbiome studies do not hold up, okay, to support their own hypothesis. In fact, recently, the same group has published a study walking back some of the association between the microbiome and various responses, to the ingestion of choline and carnitine.
Are there any other big factors that might be confounding this relationship between TMAO and disease, maybe perhaps in the study populations that have been?
Yeah, so it's a great question, and the answer is yes. TMAO is extraordinarily sensitive to even minuscule decreases in normal renal function. So in other words, give you an example, the normal renal function, a healthy adult is about 100 milliliters per minute, okay, that flows through the kidneys. When there's a drop to less than 90, which by the way is pretty normal for a lot of people in their late 50s and 60s, okay, the amount of TMAO that goes up is dramatic. It's a much more sensitive marker than even typical medical markers of renal insufficiency, which is blood urea nitrogen and creatinine. So TMAO is extraordinarily sensitive to that. And the population that was studied that was found to have TMAO elevated also happened to be in patients who were diabetic, who were smokers, who were hypertensives, who had kidney disease, who had low kidney function, much lower than 90 milliliters per minute. And as a result, you would expect to find elevated TMAO. That doesn't mean that TMAO has caused the cardiovascular disease, which is very commonly found in smokers, diabetics, hypertensive individuals. So the population that was studied in which this was brought out, okay, as proof of TMAO's role in cardiovascular disease, it's really a sick population.
No,
many of whom who'd had, 40, 50% of whom had had cardiovascular disease diagnosed, heart attacks, strokes, okay. So give me a break, okay.
Yeah.
TMAO is not a risk factor.
Yeah.
Okay, a risk factor is something that you identify in a healthy population, track, and then see that, oh, wow, people who've had this particular risk factor have a higher chance of developing the disease. They did it the other way around. They looked at a sick population, picked out TMAO, and then looked for the reason for it.
So it sounds like, a quick summary of that, I guess, would be we'll call it mechanistic issues with the hypothesis on how TMAO might impact health. There's also some confounding biases, perhaps, in some of the epidata that's out there, connecting TMAO to cardiovascular disease endpoints. And, I think at this stage it seems, difficult to really make a solid conclusion about that, or the solid conclusion that there is this significant concern. Is what I'm understanding.
Well, we have, I think, done more than our due diligence. We've also invited experts to come in. One of the areas, one of the things that's been interesting is that these studies that have published this hypothesized construct really go into multiple different non-overlapping areas of expertise that most people, very few people actually contain or have all the tools in order to understand all the components. So when we've actually spoken to experts in all of these areas, what we have found is that not only are there real challenges in the way in which the science has been performed, but so we are very comfortable that we've done our due diligence to make sure that choline is a absolutely safe nutritional nutrient. It's essential for so many things. How can it be toxic? Okay, we're suggesting it in much lower levels than what's been agreed to by the Institute of Medicine as the upper limit. So it doesn't make sense. However, in a sense, the genie's been let out of the bottle. It's not that easy to put it back in, okay, even though we're comfortable that the veracity of the science, I think, doesn't stand up to what has been claimed.
Dr. Bortz, it's obviously an important issue, and I want to thank you for your dedication to understanding and explaining the data and coming on with us to set the record straight. So thank you for joining us today.
My pleasure, Scott. Thank you.
You're welcome. All right, our next session is with Dr. Julia Maeve Bonner. Dr. Bonner has a PhD in molecular genetics from the University of Toronto, and she did her postdoc at MIT. She's now the principal scientist at Sanofi. Dr. Bonner, I got to tell you, out of all the amazing presentations that we saw at the New Directions in Choline Symposia, yours was my favorite, and I think that's probably because, it was a bit personal for me. I've got two grandmothers that passed away with Alzheimer's. My mother passed away a couple years ago with Alzheimer's, and I have a younger brother that's got the early onset version of that. So I was obviously very attentive in listening to your presentation. Found it very interesting. If you wouldn't mind, kind of just give us a general overview of kind of the high points of what your presentation was about.
Sure. First, I'm sorry to hear about your grandmother and your mother and your brother. This is why we do this work. It's a devastating disease. So I'm sorry to hear about that. I hope that we can use some of this work to move the field forward. So the summary of my research is at MIT. So in the lab of Dr. Li Weicai, I was working on a genetic risk factor for Alzheimer's disease, ApoE4. So ApoE4 is the most validated, highly validated genetic risk factor for Alzheimer's disease. Occurs in about 14% of the population, the control population, and is enriched to about 50% of Alzheimer's disease patients. And it is dose dependent. So, the more, if you get one copy of ApoE4, you have an increased risk of about 4-fold. If you have 2 copies, it's about 10 to 12-fold. So it's clearly very closely linked to Alzheimer's disease risk. And we wanted to understand, what is happening in ApoE4 compared to the non-risk or neutral risk allele rather, which is ApoE3. What's happening in the risk version versus the non-risk that's changing the cellular state, changing the brain ultimately to create this sort of early system of risk. So we were characterizing this gene and this variant of the gene, the risk version, in the cell type in the brain that makes the most of this protein. So these are astrocytes. These are glia. They support neuronal cells. They also have many functions in the brain, one of which is to sort of, it's a major modifier of lipid homeostasis. So this is the fats in the brain that are, the cell surface, the membranes of the brain, the intracellular organelle membranes, and many important components that get transferred that are important for cell function and then get transferred between cells. And ApoE, the protein, is one of the major facilitators of transport of these kind of lipids, these fats between cells. And so what we found is that in this brain cell type, astrocytes, there was a really distinct shift in the lipidome. This is all the kinds of lipids that are being made or accumulated in the cell. And the ApoE4, the risk version, accumulated neutral lipids to a much higher degree, a particular kind of lipids, especially these triacylglycerols. These are the kind of lipids that accumulate in these intracellular compartments called lipid droplets. This accumulation is associated with dysfunction of many different kinds of functions in the cell. We found that using astrocytes, as well as from work that we started initially in Susan Lindquist's lab at the Whitehead in yeast, that we were able to pinpoint a node, a biochemical node, where we could kind of influence this lipid dyshomeostasis and the resulting outcome for the cell. So we found that we were able to discover that if these cells were grown in a media that was supplemented with choline or rich in choline, compared to media that was deficient or minimal in choline, that we could really alter this lipid imbalance and move it a lot. If in the media that was supplemented for choline, move these cells quite a bit closer to the cells that were in this non-risk state, the ApoE3. We could move that lipidome, so all the kinds of lipids it's making, but especially these neutral lipids that were accumulating, these triacylglycerols, we could move them a lot closer to sort of ApoE3 or non-risk levels. And then through a lot of genetic work, we were able to pinpoint that this was actually phosphatidylcholine synthesis. So we saw that the Kennedy pathway of phosphatidylcholine synthesis was critical for this effect of choline on modifying the lipid balance. We then have been, since then, that work was published in Science Translational Medicine. And since then, we've done a lot of work to try and see, could we move this into a potential therapeutic in an in vivo and an intact animal. So we were looking at mice that have Alzheimer's disease genes knocked in. So these are amyloidogenic genes. This means that these mice have mutations, human genes with mutations in them that cause them to accumulate the plaques that we see in human brains in Alzheimer's disease. And also in the background of this, they have the human ApoE knocked in, which means that they're expressing human ApoE either 3 or 4. And in this ApoE4 background with this amyloidogenic mouse model, we fed them diet that was high or low in choline. And we saw a really nice protection against the accumulation of some of this Alzheimer's related damage, as well as a shift in the lipidome of the cells that we looked at. And some of the tissues we looked at, and we're still characterizing these outcomes, but we're excited. We think that it's possible that something that is as simple as a dietary supplement could maybe merely make a difference, especially for those who have a copy of this particular risk allele. So could be basically through modifying this lipid imbalance, we're hopefully kind of restoring a little bit of the norm or non-risk state to the cells that might otherwise kind of be in this imbalanced or risk state. So that's the gist.
Yep, that's a big picture. I'm kind of curious. So if and I'm going to ask you a question I know you don't know the answer to, but I'm going to ask you to speculate, so if you were to use choline supplementation to kind of stave off Alzheimer's, if you had the ApoE gene, would you have to be taking that or supplementing throughout lifetime? When would be the best time to be making sure you had enough choline supplementation in your diet?
So, we can't say these are mice and cells in a dish. We have human cells in a dish and they're mice. So we, of course, have to be very careful about making any claims that that this, can have the beneficial effect in humans that we would hope. There have been previous clinical trials looking at choline supplementation in Alzheimer's disease with some mixed results. A lot of those weren't stratified for ApoE status, like whether they were ApoE4 versus ApoE3. And there is one study where it does look like when they did stratify that the ApoE4 responder or carriers were responded a little bit better. But, we'd need to do a more careful, very on purpose, intentional study in humans looking at, different levels of choline with, in ApoE4 carriers, having tracked, ideally looking at sort of certain lipid markers maybe in their blood and following that, a clinical trial is required to be able to say this. And we need to be careful to make sure we don't overstate claims. But I think that, our science suggests that it should be possible and that it's worth doing these kind of studies to identify if there is, an intervention like a supplement, like food, like, eating choline-rich foods that could make a difference, because of course this would be a wonderful thing. One thing I can say is that in general, according to sort of nutritional studies, people tend not to get enough choline just by, Surgeon General recommendations. So I think that it's probably safe to say that you should get as much as is recommended currently, and then hopefully we can, do more studies to really identify if it has a specific benefit for ApoE4 carriers.
And can you talk a little bit about maybe are there future research studies planned either by yourself or others?
So there is a small study currently being planned in collaboration with MIT and the Neurodegeneration Consortium out of MD Anderson, and I think with Balchem, that have sort of been working together to try and get not a full phase 1 clinical trial or phase 2, but a clinical study in humans to try and identify biomarkers that would change on different levels of choline, and then this would create the kind of readouts we would need to know before going into a clinical trial. But of course, clinical trials are then very expensive, so we'd have to sort of see what happens after that. But this would be, hopefully a proof of principle that we can change important and relevant biomarkers, and then those can serve as readouts in a potentially bigger study.
Eric, you have any questions?
Yeah, I'm just curious if maybe you can comment on what the relationship is between, say, choline intake and neurodegeneration in epidemiological literature. Is there a connection between these two things? I mean,
so there's correlative evidence, which is that groups that are at risk for low choline, there's an overlap of groups that are at risk for low choline versus groups that are at high risk for AD. So, for instance, women are at higher risk for low choline, both from in the potential childbearing times when there's, because choline is also in the folate cycle, folate metabolism pathway. So there's a much higher requirement for choline in women, and so then you could, they are potentially at risk for being older people. It's been reported that the elderly, there's a decrease in their ability to uptake choline, even to whatever level that they have it circulating from their diet, that there's a decreased ability to take it up. So this could also increase risk, and of course we know that age is the biggest risk factor for AD. And there are some studies that suggest that AD patients are have lower levels of choline, but it's not super consistent. So that would, those studies, it kind of depends on what's being measured.
Definitely an interesting emerging area for sure. Really neat to see sort of some of this pioneering work you've done mechanistically. I think the picture is very complex. I mean, as you said, this relationship between ApoE4 and other dietary factors is tricky, and I think something that's only been appreciated in the last, I don't know, let's say 10 years or so, really, to a certain degree. I mean, the reason I love your work in particular, one of the reasons, is because I do think this appreciation for specific genetic markers and how specific dietary interventions might help people with those genetic markers, I think is incredibly important. We're definitely at a stage, I think, in the field of nutrition where a lot of the sort of big picture general population recommendations have definitely been covered. And I think this area of nutrigenetics, precision nutrition, personalized nutrition, whatever buzzword you want, is definitely the future. So I really
well, thank you. And I think I agree. I think that, similarly from, a field of Alzheimer's research, we've done a lot where we try and look at sort of a broad scope of Alzheimer's disease suffering patients and compare, all AD against all control. And, the more we learn, the more we understand that there might be subtypes within the disease. There might be, within that all kind of converge on a clinical presentation of memory defects and neuronal loss. But, there might be underlying, mechanisms that are slightly different or have slightly different sort of origins or starting points. And if we can identify that that's the case, if we understand whether or not that's the case, and then we can potentially understand that there are more targeted therapies that might be more amenable to some AD patients versus others, and that if we understand sort of how they're getting to their clinical presentation, we can have a more effective series of therapies.
In your animal study, do you know what the equivalency would be to a human diet? For example, in the supplemented mice, were they getting?
So the choline dietary recommendations for mice are very high, much higher than for humans. We went basically our dietary paradigm. We wanted to kind of try and keep as, within the scope of what sort of healthy mouse would see. So we went to sort of the National Research Council minimum and maximum, and those were about fivefold different between the minimum choline and the maximum choline, about fivefold different amounts of choline. But those are much higher. They're in grams per kg amounts of choline in the diet compared to 450 mg per day that is recommended for adult males or adult females. 450 mg per day, somewhere in that range. There are much higher requirement in mice, so we were within the sort of healthy mouse range, and then a fivefold increase in the supplemented diet.
I do think your comment is very prudent, right? I mean, I think what we know, of course, that before making any kind of strong claims about the connection between choline and the disease, of course, we need rigorous RCTs. But I do think as a prudent dietary piece of advice, it's probably smart to at least stick to trying to get your daily requirement. So I would agree. I think you're spot on with that recommendation. Just tough. Not a lot of people do that, right? I think you said depending on the source, it's what 6 to 10% of people get enough. I mean, it's really not a lot. So a lot of work to be done in terms of education and awareness.
That it's tough to get the appropriate dietary amount of choline. So humans do make some choline, but not enough. It is a required nutrient. So it can, it's depending on your diet. I think, the recommendations that are always the case for most diseases of aging, most healthy living in general, which is, a good balanced diet and taking care of yourself in every way that you're able to, are always safe recommendations. But again, in terms of specific recommendations for, choline intake beyond the already, we would need to do the careful studies. But yeah, most people don't even achieve that much. So so it seems prudent to watch what you're eating and do your best.
Dr. Bonner, as we get ready to close out here, any key takeaways you'd like to leave with our audience?
Well, I appreciate, that this is one of the sort of newer angles of looking at Alzheimer's disease is looking at it from a sort of metabolic, potentially nutritionally accessible viewpoint. I think one of the key, one of the things that will help, I think, to understand different kind of interventions and how they can be most helpful is these kind of mechanistic studies, these kind of this work where we can understand, there are broad benefits to the things that we know our body needs, but if we can actually, start linking those to specific pathways, to specific mechanisms, to specific, requirements that may be greater or lesser in certain risk states, then I think, there's a much greater case. It's much easier to convey that information to a general public, but also hopefully to convince, the medical field that these are valuable insights. And of course, with that, I just would also like to say that all this work is, it's publicly funded. We are excited to share it. We hope that people can learn from it. And, the most important thing is if we can try and make a difference for people who are suffering from this terrible disease.
Yeah, well said. I want to thank you for your work on this important subject, and I want to thank you for joining us here today.
Yeah, happy to.
Appreciate it.
Thank you.
Tonight's last call question is brought to you by NitroSure Precision-Release Nitrogen. NitroSure delivers a complete TMR for the rumen microbiome, helping you feed the microbes that feed your cows. To learn more about maximizing microbial protein output while reducing your carbon footprint, visit balchem.com/NitroSure.
Well, folks, we just concluded the future directions in choline. I hope you've been able to join us for all of the interviews so far. If not, please go back and listen to the first one. We started off the conference with Dr. Stephen Hursting and Dr. Susan Smith, both with UNC here. And we asked you, what does success look like? And so I'm not going to ask that exact question, but was it everything you expected?
It was more than I expected, actually. So we had more like 90 people here. One thing we were really looking forward to was showing our campus off and our facilities. And so that was so much fun for us to do. But also the science was just unbelievably good. And the progress that has been made we talked about this 25 years since really choline was established as a nutrient. The progress over that 25 years is remarkable. And we heard that so many exciting talks. And to me, you expect that from some of our senior folks that were here. To me, the highlight was our emerging stars and the excitement they have in this, the new ideas that they're bringing to the field. The field's in fantastic shape, and so that was really exciting. So I think I said we were looking at what would be a touchdown, what could we spike the ball on? Well, I think we scored 8 touchdowns in this event.
Well said, Susan. Based on what you heard this week, so what are the future directions of choline?
Yeah, so it was interesting reflecting upon where we were and where we are now. And so we came into the conference, what had set up choline as an essential nutrient was the fact that it promotes liver health and it promotes skeletal muscle health. And what we heard about this time was we saw tremendous cognitive effects when given during pregnancy and early childhood. It's building smarter brains in babies. We learned about a potential role in Alzheimer's disease. We learned about roles in, again, liver health, but also its potential for obesity and helping to overcome some of the side effects that the ill health that can accompany obesity. Just so much exciting information.
Yeah, I couldn't help but reflect on after hearing a lot of these presentations and all the amazing benefits of adequate choline supplementation. Why is it so? Why is it the nutrient nobody's heard of? Right? It's just that's a little confounding to me. I don't know if you guys have an answer for that, but
One thing I learned is that there haven't been many opportunities for the broad field to get together. It's been in various, those that work on early development, maybe go to pediatric meetings. Those that work on later in life are going to more the aging or the Alzheimer's types of conferences. But this may be the first time for many in the field that have gotten together with the whole breadth of the field. And so I think that's, we're going to do it again, by the way, in 2 years. I made the commitment that let's start this party again in 2 more years and invite some of our friends at NIH, FDA, some of the other decision makers in the area of nutrition and disease health, promotion, to come and listen to this progress. And so we're looking forward to that already.
And if I might, I'd please invite some of our researchers in animal nutrition.
Yes.
There's a lot of bright people out there doing some pretty cool stuff. Now, it's not exactly what you guys are doing, but I think there might be some synergies there where you can learn from each other.
100%. We had actually a couple of veterinary medicine researchers here that shared some of their results in the animal health area. Fascinating, and I think that's exactly right. We'll have a whole session, I think, around that topic next time.
Yeah, perfect.
Yeah, I'm really excited about the opportunity to come back in 2 years, I think, and show all the progress we've made. Obviously, there's been a lot since 1998, but I think back even 5 years ago, we held a roundtable with some of the leaders like people from NIH and from USDA. And I remember one of the fellows putting up on the screen, a picture of choline with 500 miles to go before getting to the science that they needed for a DRI. And I think in 2 years, when we bring those guys back, we're going to be able to show that we've been driving through the night. We're a lot closer than they think.
That's a great way to put it. I love that.
Yeah. I think that's true, because when we were organizing the conference, it was so hard to pick and choose what work we could feature. We could have filled a week easily. And so in 2 years' time, it's going to be really exciting to hopefully we can expand this a little and hear even more about it.
Yeah, super.
I also love the opportunity, and you brought it up, of the young scientists to present. It was great to see the posters and really celebrate some of the up-and-coming work. So lots to build on and more exciting things to talk about.
One of the things you did right at the end of the conference is you had these breakout sessions where everybody got together and was sharing ideas, and there was a lot of sparks flying and innovation going on. I really enjoyed listening in on those. Can you share some of the ideas that the teams came back with?
Eat more choline.
that was a theme.
I think one unexpected discussion circled around the effects of the climate change and heat stress and the impact that could be having upon both choline needs as well as the choline content of foods. We know that it's having an impact, say, upon the protein content of certain plants and the like, but we don't know what that impact is going to be with choline. And so there's an area of research to think about.
Wow, yeah.
Interesting.
I was struck by I think it's a sign of the maturation of the field of efforts to really align research and standardize, really, so that data can be pooled in future and get more powerful summaries of data, such as meta-analyses, where you can put multiple studies together and get a much stronger signal than a single study. And so there were efforts in that regard of aligning on biomarkers, on standardizing dosing and type of choline administered, and study design, so that those types of pooling efforts can occur in a few years. And I think that will really move things forward. So I was struck by that.
Yeah.
Well, on behalf of the Balchem team, because it wouldn't be fair to close this without saying we really appreciate all the leadership from UNC and the Nutrition Research Institute over the past years, and we're really excited to have started this ball rolling and to continue it. So thank you so much for hosting us.
Yeah,
Thank you very much.
Yeah, well said, Tom. One final question for each of you guys. Dr. Ciappio did a nice job of summarizing things there toward the end, and what he did was he asked everybody in 2 to 3 sentences, tell us what the key takeaways are. And so, use this opportunity, right, to share this succinct message to a wider audience. So I don't know which of you guys would like to start, but
I'll say choline looks like an easy way to work toward a healthy lifespan, and not just a lifespan, but a health span. It's touching everything from fetal development to early childhood, adolescence, adulthood, and into aging. And it's just such a huge spread and an easy way to address that. Eat more eggs, take more choline.
Well said.
Well said. I can't add anything to that. Perfectly said.
So we're going to leave it there. All right.
Thank you very much.
There you go, folks. Eat more choline.
Eat more choline. That's right. That's exactly right.
Well, listen, this has been a great conference. I want to thank you guys for putting it together. Thank you for joining us. This has been a blast. Also want to thank our loyal audience for joining us once again. I hope you learned something. Hope you had some fun, and we hope to see you next time here at the Real Science Exchange, where it's always happy hour and you're always among friends.
We'd love to hear your comments or ideas for topics and guests, so please reach out via email to anh.marketing@balchem.com with any suggestions, and we'll work hard to add them to the schedule. Don't forget to leave a 5-star rating on your way out. You can request your Real Science Exchange t-shirt in just a few easy steps. Just like or subscribe to the Real Science Exchange and send us a screenshot along with your address and t-shirt size to anh.marketing@balchem.com. Balchem's Real Science Lecture Series of webinars continues with ruminant-focused topics on the first Tuesday of every month, monogastric-focused topics on the second Tuesday of each month, and quarterly topics for the companion animal segment. Visit balchem.com/realscience to see the latest schedule and to register for upcoming webinars.